My name is Charles Manning from MD Anderson Cancer Center in Houston, Texas, and it's my privilege to introduce my co-moderator, Mei Shan, who is our WMIS president. She'll introduce Dr. Evans.
Hello, good morning. My name is Mei Tsien from Fudan University. I would like to introduce Dr. Michael Evans. He is an associate professor in residence in the Department of Radiology and Biomedical Imaging at UCSF. He is a chemical biologist with an interest in biomarker discovery, radiopharmaceutical development, and translation to study oncogene signaling in cancer, and early drug development for nuclear theranostics. Dr. Evans has earned his BA in chemistry from St. Mary's College of Maryland, and he obtained his PhD in organic chemistry from the Scripps Research Institute, CA, under the supervision of Professor Benjamin Karavat. This was followed by a postdoc fellowship in molecular imaging from Memorial Sloan Kettering Cancer Center in New York under the supervision of Professor Charles Sawyer and Jason Lewis. In 2013, Dr. Evans accepted his faculty position at UCSF. So today, it's our great pleasure and honor to invite Dr. Evans to come to the stage to give the award lecture. Dr. Evans.
Let's have a warm welcome.
Okay, thank you. This is a huge honor to be associated with, there we go, Professor Roger Tsien. Today, the title of my talk is Chemical Strategies to Maximize the Anti-Tumor Effects of Targeted Radiotherapy, but it's really kind of a love letter to the nuclear medicine and chemical biology communities to try to underscore how there's mutual synergy at the interface of these two communities and try and hopefully encourage them to continue to work together. Before I start, just a brief comment about Roger Tsien. Like I said, it's really fantastic and quite an honor to be associated with him in any way. It was a huge luminary in the chemical biology field and one of the first, in my mind at least, crossovers into the molecular imaging field. Of course, everybody knows him for GFP, but a little bit later in his career, he started getting into, more medical applications of imaging. And this paper I highlight because it's just, it's one of the first encounters that I had with a molecular imaging paper that really got me thinking about the field. And so, all these years later, it's somewhat, I guess ironic in a way that I'm standing up here, receiving his award. So targeted radiotherapy, just as a level set introduction, like I said, today's talk will be mainly about what I think are, chemical or kind of chemical biology-ish strategies that we can use to potentially augment the effects of this really, really promising therapeutic modality. And I think as many of you know, this fundamentally is a modality in which, a systemically administered drug is trafficking ionizing radiation to the tumor. And I like to think of it almost as, a way to take the well-established virtues of radiation therapy and mobilize it to a setting where external beam radiation therapy is just not practical, in particular, metastatic disease. And so I just highlight this review because it very nicely, or this figure from this review, because it very nicely underscores the various different mechanisms by which one can use to traffic ionizing radiation to the tumors. And I think it's fair to say that the field is, probably most bullish on low molecular weight radioligand therapies at this time. So that being, say, maybe a small molecule, peptide, nanobody, what have you, that targets some protein, typically on the outside of the cell, not necessarily, that's highly overexpressed in cancer versus normal tissue, thereby achieving the therapeutic index. And with good reason, we should be bullish on this, right? Because there's been a number of FDA approvals in the targeted radiotherapy space, two of which are radioligands in Lutathera and PSMA. So there's clearly some value to radioligand therapy, and I think, more to come as money flows into the space and there's an increase in clinical activity. But, when you look at the statistics, at least from the data that we have right now, it's not always a very impressive effect size, So these are just overall survival curves from the VISION trial with lutetium PSMA-617, and it's about a 4-month survival benefit. By comparison, enzalutamide, abiraterone, those are about a year to 16-month survival benefits. So this is a little surprising to me, because there's no questioning the virtue of the payload, right? We understand that ionizing radiation is a very, very effective way to ablate tumors. There's very few resistance mechanisms that we know that are truly innate resistance mechanisms. There's plenty of things that cancer cells will do to adapt to ionizing radiation, but it's not permanent. These are transient, kind of maneuvers to try to outlast the ionizing radiation. But then also, we have this theranostic paradigm where we can actually pre-screen patients with imaging to drop out those who just lack target expression. That's an extremely powerful way to denoise the clinical data. And in spite of all that, we get fairly, at least to date, meek survival benefits. So that to me seems like an unmet need. And in chemical biology, one of the things that we do very well, I think, is thinking creatively about early stage drug development, both on the biomarker side and then also on kind of the drug design side. And so I just asked this, semi-rhetorical question, what are the unmet needs for the nuclear medicine field or targeted radiotherapy field to which the chemical biologists can contribute? There's lots of answers to this question. The two areas that we think about most in my lab are first the delivery, right, coming up with different styles of drugs to increase the efficiency of radiation delivery to tumors, without necessarily playing the serum half-life extension game. So with a minimal or no impact on the pharmacokinetics of the drug, how can you actually get more radiation delivery to the tumor? And then, of course, new tumor-specific targets, which have been very elusive for this field. So why should we try to kind of outdo something that at least today with the state of the art is working? And I would argue that there are some intrinsic challenges with RLT that we're never going to get around unless we step out of the RLT paradigm. One of them is, ligand receptor interactions in biology are just transient. Mother Nature knows how to destroy ligand receptor interactions, and we can see that very clearly with just longitudinal imaging of things that we know work, so to speak, like lutetium PSMA-617. And there's lots of different mechanisms by which ligand receptor interactions can be broken down, including the nuclear option where we just destroy it in the lysosome. The other issue is that there are very few proteins that actually meet the criteria in biology to be an RLT drug target, probably fewer than 100. And so we're going to hit a wall, I guess is what I'm trying to say. Tumor heterogeneity is a blind spot in the radioligand therapy paradigm. And we basically have kind of put ourselves into a model where we treat the cells that express the target and really no other cells, with the caveat that there are some crossfire effects that can be associated with the payload. There's also kind of like an unusual safety-efficacy trade-off that we deal with in targeted radiotherapy because of the nature of the payload. So the payload continuously emits. We can't mask an isotope and then conditionally release it in the tumor microenvironment. So, one of the easiest ways in medchem to increase tumor exposure of a drug is just manipulate the half-life of the drug in the serum, right? But when we play that game in targeted radiotherapy, you induce collateral damage. So if we were to do this with like an ADC, the MMAE basically stays tethered to the antibody. Long circulation time isn't a consideration at all. But if you pivot to something like lutetium, longer circulation time can lead to undesirable tox. So in the spirit of this conference and the theme of, what if, I would say, well, what if we can step out of, the current status quo, this trade-off here, and arrive at this type of ideal new treatment where you have, rapid clearance from circulation to get a desirable safety profile, but some sort of chemical trick installed in the tumor to get an AUC that's a little bit more like a high molecular weight radioligand compared to a low molecular weight radioligand. And so, how are we trying to do that? I'll just talk very briefly about two different approaches that we're using. One is by, basically leveraging a platform that was really developed for imaging a little under 10 years ago by my colleague Charlie Craik at UCSF, who's a protease biochemist. And he developed this platform called a restricted interaction peptide, basically to try to measure enzyme activity in vivo using imaging. And the way the platform works is that essentially you administer this pro-peptide, a linear, about 4 kilodalton peptide that has an unstructured conformation. And when the peptide encounters a protease of interest, an endoprotease of interest, I should say, there's an internal cut site which releases this masking domain and allows the blue domain to adopt a helical conformation that then can cause membrane insertion. And so these are just actually like antimicrobial peptides. They have a well-documented history of interacting with membranes. They run the gamut from things that can puncture membranes to things that can actually just kind of lay down and nestle in the membrane and not disrupt it. And we have a tendency to work with the ones that don't cause membrane lysis. And so our thought behind why these could be potentially useful therapeutics, really came just from the initial tumor imaging data that we gathered and recently published, with a so-called RIP targeting the secreted protease granzyme B that's, pushed out of activated immune cells as part of, anti-tumor immunity. And so don't want to go too deeply into the story here, but basically there are two pieces of imaging data that we observed that gave me some hope that there could be a potential therapeutic play for a different type of RIP. And the first one came from this longitudinal imaging study that we published showing that the RIP gets into tumors very quickly, but it also just like hangs there for a very long period of time. We only presented 24-hour data here because we weren't making a big deal about the residence time in the tumor for an imaging paper, but we actually carried these imaging studies out to 96, 120 hours post-injection, and you can see that the antimicrobial peptide is just latched and anchored into the tumor. And this does make sense. I mean, we produce antimicrobial peptides without inducing an immunogenic response. There's just not a tremendous amount of evolutionary pressure for cells to get rid of something that's benign and innocuous like an antimicrobial peptide, and in some settings it's actually very helpful just to keep around to clean up pathogens. The other thing we really liked about this approach is that when you look at digital audio radiography, you can see that the membrane-inserted peptide really kind of smears across the tumor and extends well beyond the boundaries of granzyme B on immunofluorescence. And that suggested to us, and we don't really have proof that this is happening, but it suggested to us that we might be getting this kind of effect with a RIP, which could enable us to actually label cells that don't express the target. In the case of the granzyme probe, none of the cells express the target, it's secreted. But if we were working with like a membrane-anchored protease, then potentially we could actually smear into regions of the tumor that lack the actual protease, which would provide some mechanistic disruption from the RLT paradigm. So I'm going to show a little bit of redacted data here in the sense that we're not going to talk about what actually the protease is that we're targeting, but this is one of the first examples of a RIP that we developed specifically to be a drug, a targeted radiotherapy, just calling it UCSF01. Because of the nature of the protease that we're targeting, we could run a head-to-head comparison between an RLT targeting the same protease as this RIP that's activated by the protease. And what we were able to observe in these mouse subcutaneous tumor models is that you basically win on both the Y and the X axis. You get higher tumor uptake than you get with the RLT, presumably due to catalytic amplification of the activation of the probe, but you also appear to get longer retention of the probe in the tumor. And importantly, all of this comes with really no impact on the ligand pharmacokinetics. So this is just a dynamic scanned blood curve. You can see that the serum half-life of both of these reagents is very quick. If you payload it with a therapeutic, it delays tumor growth. And so in this case, we just chose copper-67 because it's very similar to the copper-64 we were using. Single injection, high-dose injection, you can see you retard tumor growth. There's a survival benefit, and the drug is well tolerated. And so again, to summarize, we think based on the data, what's going on is this effect where, we see some gain beyond the stoichiometric limits of the RLT, the ligand receptor complex because of the renewable nature of the activation mechanism, but then also a longer residence time in the tumor because we're anchoring in the membrane and not dealing with a ligand receptor interaction that the cell knows how to break. And for what it's worth, we just got our study may proceed letter for the first imaging granzyme probe, and we're in the process of recruiting for the first in-human cohort. Okay, so the second vignette I'll talk about is not so much about increasing uptake of the radiotracer on the y-axis in the tumor, but rather about increasing the residence time of the radioligand in the tumor by engineering covalency into a biomolecule. And so the backstory here is that there's one of the big success stories in chemical biology over the past 30 years has been the development of unnatural amino acids and incorporating them into the genome. And so there's a whole variety of unnatural amino acids that can be engineered for a variety of different applications. My colleague at UCSF, Lei Wang, has been a pioneer in installing electrophilic amino acids with electrophilic side chains to try to engineer covalency into biomolecules. And so we started collaborating a couple of years ago to ask the question, if one were to engineer covalency into a targeted radiotherapy, would you actually get better anti-tumor effects because you've essentially destroyed the K off by creating this covalent link? And so using HER2 as a model system in nanobodies, we basically developed this pair of nanobodies that differ by a single amino acid, just the FSY substitution compared to a tyrosine in the wild-type molecule. Doing imaging studies with, kind of an imperfect radiolabeling strategy of just iodination, nevertheless shows that you get an increased resonance time of the mutant antibody in blue here in the tumor compared to the wild type, which actually loses radioactivity pretty quickly post-injection. And these are just some maximum intensity projections showing that, on a normalized scale, you can see persistence of the radioactivity in the tumor, set to a gating where you can't really see anything happening with the non-covalent nanobody. Again, we're trying to do this without manipulating PK. And looking at the blood curve data, you can very clearly see that these are almost indistinguishable in terms of serum half-life. And that makes sense. It's a single amino acid substitution. And then if we pivot and payload the nanobodies with an alpha emitter instead of a PET emitter, you can actually see that it translates into better anti-tumor effects. And so, this is still a work in progress. We're not making tumors shrink and go away. But with this type of experimental design, you can very clearly see the difference in the effect size between what the actinium-labeled non-covalent nanobody is doing versus the covalent nanobody. And this is all just done in a subcutaneous mouse tumor model. For what it's worth, there's a company in this space that's commercializing this technology, which I think is always exciting to take things, out of the lab and into the clinic. And so, they've, coined this approach, the Warlock technology. But basically, one of the top things on their list to do in LASA is to bring covalent biologics in the form of radioligand therapies into patients. And so, this is a company that's in San Diego that recently came out of stealth mode with a pretty significant Series A raise. And so the second bullet point I'll talk about is new tumor-specific targets in the last 15 minutes or so that we have for this talk. And so what's the unmet need here? And I think I put this up to be constructively critical, not to be unconstructively critical, I guess I would say. When I started thinking about targeted radiotherapy and what could I bring to the field with my experience, immediately one's mind starts drifting towards what are the kind of bottlenecks in this space, and one of the things that I think we really struggle with that frames the rest of the conversation is picking the right drug target from the outset, and just doing kind of an audit of the clinical activity in this space going back just about 1 or 2 years ago. So these data are slightly out of date. You can very clearly see that if you've been in clinical trials by target, about 40% of all the clinical trials that are going on are focused on about 2 targets. Today the somatostatin receptor has probably crept up a little bit higher on that list. So we probably have a, kind of a 3-headed beast of PSMA, FAP, and somatostatin receptor radioligand trials ongoing. But the point is that I think we're not sampling a tremendous amount of biodiversity, and there's really not a compelling scientific reason for that other than, we're just not searching for new targets. And so, at Scripps, I was, very heavily involved in proteomics, and so when I came out to UCSF to start my lab, the first thing I thought of doing is, just basically a type of proteomic profiling that's biased towards identifying targets that would be useful for nuclear imaging and/or targeted radiotherapy. And I very quickly started collaborating with Jim Wells, who is, kind of already doing this, but with an eye towards other applications outside of nuclear medicine. And, the basic premise here is that, one can, really denoise the proteomics data through the careful design of the upfront profiling experiment. And so what we're trying to do is basically identify protein targets that were induced in a bad cancer genotype versus a benign cancer genotype, rather than just kind of, screening for things that are upregulated in melanoma versus breast cancer. And so we engineered a whole panel of human isogenic pairs with, tumor suppressors or oncogenes installed, that are very visible and common. Ran some, cell surface trapping experiments, shotgun proteomics to quantify things, and then there was a handoff. And a lot of these targets were worked upon or put through a phage display platform to identify antibody-based binders, but in some cases we also developed some small molecule peptide binders. It was a very, very fruitful kind of 8-year run, I would say. And this table is just basically kind of summarizing all, some of the higher profile publications that have come out of this workflow, where we go from proteomic screen, hit identification, identify a ligand, and then payload it up and show some utility in imaging and/or targeted radiotherapy. What I would say, though, is that my takeaway from all of this is that we have yet to identify a truly tumor-specific target by using this type of abundance-based screening. So we've identified a lot of really interesting targets, like, for example, this guy, Cub domain containing protein 1, which is massively overexpressed in a lot of different tumor types, but has that, kind of caveat that it's not truly tumor-specific. And so we're starting to think about it in the other direction. Should we instead be prioritizing selectivity for tumor over highly abundant tumor targets that aren't particularly selective for tumor versus normal tissue? And part of the motivation here is, the emergence of alpha emitters, right? With alpha emitters, you don't necessarily need 10 to the 6 copies per cell to destroy a tumor. So this represents, kind of a conceptual pivot for us. And I'm just going to very briefly, kind of highlight some of the, the redacted data and confidential data that we have in this space. But, the idea here is, let me pull back and say, this really started as a collaboration between myself, Charlie Craik, Kayvon Shokat, and one of Kayvon's graduate students who now runs a lab at Berkeley, Ziyang Zhang. And the question that they were interested in asking is, can one actually demonstrate that the ultimate fate of a labeled oncoprotein is at least in part displayed through the MHC class I pathway? And if that is present on there, can you target that signature molecular lesion with a very highly potent and specific biomolecule, an immunoglobulin in this case? And so that's the general concept. The way this was reduced to practice as proof of concept was by looking at KRAS G12C inhibitors like ARS 1620, treat G12C positive tumors, allow the covalent labeling to take place, and then just kind of let the cells percolate for a while and see if they display a sufficient amount of the haptonated peptide on the surface of the cell to be detected with an antibody that was evolved to bind this complex selectively ex vivo, outside of cells. And so Charlie, Kayvon, and the team published this Cancer Cell paper in late 2022, and since then we've been evolving second and third generation antibodies that do slightly different things and running them through in vivo studies to demonstrate really for the first time that a chemically induced receptor can be visualized with immunopet, which is the data that we're showing here, and I'll just walk you through it very quickly. So these are mice with subcutaneous G12C tumors. The mice are separated into treatment arms receiving either vehicle or a dose of a covalent RAS inhibitor. We pre-dose these animals so that we can get the receptor displayed. And then we administer a radiopharmaceutical and just kind of watch over time to see if the presence of the drug redistributes antibody into the tumor. So for two different tumor types, both G12C positive, you can see just by looking at the the positioning of the arrow on the tumor, that there's a relative increase in radiotracer activity in the tumor that's driven by pretreatment and continuation of treatment of these animals with a RAS inhibitor, covalent RAS inhibitor. We followed up on this, and we're now doing treatment studies, and I say without showing the data, everything looks very promising, and we're in the process of writing all this up. So for us, this is a truly tumor-specific receptor that we're targeting, which I think is important. But part of the greater clinical significance is that the patients that are on KRAS G12C inhibitors and fail because they acquired additional mutations, not because they lose G12C. And so the ultimate goal here is to translate, a payloaded antibody of some ilk, radiolabeled, CAR, ADC, whatever, into patients that have failed G12C inhibitors, and we, failure we're defining radiographically, and we keep these patients on drug, and then just begin to co-administer this additional antibody to come in and ablate what's ultimately an extremely aggressive, untreatable disease. Okay, and so with that, I'll pull back and start to wrap up. I mean, I think in my career, what I've been trying to do is serve as a bit of an ambassador between both the chemical biology and the nuclear medicine communities. I give basically two different types of talks, a blue talk and an orange talk, essentially. The blue talk reads like, what we talked about today, where, I'm kind of saying like, as a chemical biologist, there's a role for us in the early drug development space in targeted radiotherapy, and hopefully, more engagement with the chemical biology community will come around as, targeted radiotherapy becomes mainstream as a cancer treatment. To the chemical biology community, I say, why do you need nuclear medicine? And I have this, interesting and somewhat frustrating 10-year-long relationship with PSMA and the androgen receptor, and left the slides out of this talk, but basically the short version of this story is, we were searching for a biomarker for the androgen receptor many years ago, back when we were trying to understand how enzalutamide and abiraterone worked in patients. And so, we somewhat opportunistically identified that the androgen receptor will regulate cell surface PSMA expression in kind of an inverse way. So high AR, low PSMA, low AR, high PSMA. We published that data back in about 2010. A lot of people in the field got very excited by it and reproduced the data in mice. And so I have a nice table showing, the dozen or so publications that came after ours that basically all arrived at the same conclusion that we did, using different models and different androgen receptor modulators, that there's this weird inverse relationship between PSMA and AR. When I showed up at UCSF, I won over Rahul Aggarwal and Tom Hope to starting an investigator-initiated trial looking at gallium PSMA 11 patterns pre and post androgen receptor modulation. So one of the first patients that we looked at, and we published this N-of-1 patient study in the Journal of Nuclear Medicine, was a patient with locally advanced prostate cancer that was defined as such using PSMA PET, and then he immediately went on androgen deprivation therapy. We brought him back on the scanner. He had this beautiful bloom of PSMA levels in the tumor, and every tumor in his body had a relative increase in PSMA expression. It was quite remarkable. The 10 other patients we scanned after that was a total mess. The PSMA, the post-treatment PSMA levels were all over the place, and it was a little frustrating, but the takeaway is, I think, we learned more from scanning, fewer than a dozen patients, human patients, about the regulation of PSMA and prostate cancer than we learned from a decade of mouse studies. And so that's the point that I try to bring back to the chemical biology community. As you can see from people like Roger Chen, they are engaged in imaging, but IND is not part of their vocabulary. And so that's the point that I'm trying to make, is that, trouble yourself to work with modalities like PET, that while they're, they can be inconvenient, come with a lot of regulatory guidelines and safety considerations, nevertheless scale vertically into people, because I'm convinced that there's many layers of complex human biology that we're probably not going to figure out just by studying mice and rats and cells. And so I will continue to preach that, frustrating though it may be, to see, all of the model that you work so hard to establish in mice, completely rewritten by a handful of patient images. And so with that, I'll stop and do some acknowledgements. Jason Lewis asked me earlier this week, tips for young investigators. And, the immediate thing that jumped into my head was, never be the smartest person in the room if you want to, be successful and grow as a person. And, with this team of mentors here that I had the privilege of working with for about, I guess 15 years, I was certainly never the smartest person in the room, but I learned a lot and grew quite a bit. And I'm very thankful to all of them, particularly Charles and Jason, who really turned me on to imaging. I was a little imaging curious towards the end of graduate school, but it was really, Charles who pushed me towards nuclear medicine, and then Jason who really kind of like supercharged my understanding of it, and here we are. Fantastic collaborators at UCSF that helped make all this happen, and of course, the funding, and then on the last slide here, since there's a little bit of a look back period with this type of lecture, I just want to thank all of the mentees that I've been able to work with over the past 10 years, current ones, like these superstars here, and then former ones that have gone on to do, many fantastic and wonderful things, and including, picking up some of these nice awards, from international societies. And so with that, I will stop, and thank you for your time.
Let's thank our speaker. Thank you. Well, thank you again, Mike, for a wonderful session. We would like for the audience to hold questions and engage Mike after the session. We're going to take a quick picture, and then we're going to move quickly into our next plenary. So let's thank Mike again while we take the photograph.
Okay, good morning everyone. So we would like to move this session on, and we would like to proceed to the next stage, and that is my greatest pleasure to actually welcome Professor Stephen Russell here as one of our plenary speakers at this conference. Steve, if you would please come up. Professor Stephen Russell is the co-founder and the CEO of Vyriad currently, but Steve looks also back to a very, very distinguished career. And Steve received his medical doctor from the University of Edinburgh, that's up in Scotland in the UK, in 1982. And he specialized at the time in clinical hematology. He moved on down to London to the Royal Marsden Hospital, which was part of the University of London at the time, and he was the first there to engineer both retroviral and parvoviral vectors to deliver interleukin genes for cancer immunotherapy. That was long before immunotherapy was actually kicking off and became what it is nowadays. Steve then moved to Cambridge, where he led a research team at the renowned MRC Laboratory for Molecular Biology, and founded Cambridge Genetics, which is a biotech drug discovery company. And in 1998, something very important happened. He received a call to the US, to the Mayo Clinic in Minnesota, and he founded the Department of Molecular Medicine there, which is the department that you see in its current form. At that time, in the following years, he orchestrated a lot of things. He contributed to the GMP manufacture, the preclinical pharmacological and toxicological testing, the IND filings, and the clinical translation of oncolytic measles and vesicular stomatitis viruses. And very important for us as a society, he did so with imaging in view, and he actually incorporated NIS as a reporter gene, and I'm sure we will see some of those data today. So Steve was then named the Richard O. Jacobson Professor of Molecular Medicine at the Mayo Clinic in 2006, and he is also the past president of the American Society of Cell and Gene Therapy, and he's a founding member of the European Society of Gene and Cell Therapy. So please join me in giving him a welcome applause, and we're looking very much forward to his lecture.
Thanks, Gilbert, for that very kind introduction. And it's an honor and a privilege for me to be here today and speaking to you about NIS reporter gene imaging to accelerate the development of genetic medicines. So I do have a disclosure. I'm the co-founder and CEO of two companies, Imanis Life Sciences, which has historically been developing NIS imaging materials, and Viriad, an oncolytic virotherapy company. So the outline of what I'm going to talk to you about today is shown here. I'm going to start just kind of doing some brief background on the pharmacology of genetic and regenerative medicines and how we approach that, and then talk about NIS versus optical reporter gene imaging as a useful tool to deploy. And then I'm going to give specific examples that address PK/PD questions with NIS reporter gene imaging. I'm going to talk about AAV vectors and how in mice we've been able to look at biodistribution and durability of gene expression, in how we've applied that in dog to optimize gene delivery to the heart. I'm going to talk about oncolytic viruses and how we've used reporter gene imaging in mice to help clarify mechanisms and kinetics of intratumoral spread. And then in humans, how NIS imaging has helped us understand the heterogeneity of infection and the independence of infection from tumor response. There's been a big question about iPS-derived gene-edited stem cells. Well, any iPS-derived cell, as to whether or not, if it's used to repair a cardiac defect, does it actually continue to persist in the heart? So I'll talk about NIS imaging to address that question. And then I'll talk about monitoring liver regeneration using lentiviral vector transduced hepatocytes in a liver regeneration model. And then last, I'll talk about how we're currently using NIS gene transfer and imaging to monitor the dynamics of in vivo generated CAR T cells. So just here's the challenge, trying to map the in vivo fate of genetic medicines. You know, with gene therapy and gene editing and other genetic medicines, this is the big challenge of how you actually monitor in vivo in human subjects the persistence, expansion, dispersion, disappearance, and the changes over time that are happening with gene expression. That's primarily the pharmacokinetics. You know, typically with small molecules, it's absorption, distribution, metabolism, excretion. But then if you don't know the pharmacokinetics, then you can't understand the pharmacodynamics of how the drug is actually hitting its target. And really, with these genetic medicines, you've got two approaches possible. One is reporter gene imaging. The other is to do repeat multiple biopsies of multiple tissues, which obviously you can do in animal studies, but it's kind of unappealing in human. So if we look at the different types of reporter gene imaging available, obviously there are quite a lot, but here I'm just comparing the NIS radio tracer versus the luciferase optical imaging. And I think really the advantages of the radio tracer imaging, I think, are fairly clear, that it's first non-immunogenic, and there are NIS genes you can use from a whole variety of different species, and it seems to be the case, though, that you don't actually need to fully match the species. So we've not seen immune response against human NIS in mice or dogs, for example. And in fact, what we're finding is that the whale NIS is the one we prefer to use, the NIS gene from the minke whale, because it seems to give better imaging with tetrafluoroborate. As I'll talk about later. Deep tissue imaging, so that's immunogenicity. Getting into deep tissues is important. You can see from that luciferase image, you can see a blob on the surface of the mouse, but you don't know which organ it's coming from. I mean, you can guess it's probably the heart, but you can really see that beautifully when you do the NIS imaging with radioisotopes. High resolution, the resolution's getting better and better all the time with radioisotopic reporter gene imaging because the instruments get better and also the generation of high purity radioisotope tracers is getting better. And then you've got this tomographic 3D option, which, you know, people are making some headway with that in terms of luciferase, but really it's never going to be applicable in larger animal models or in human. So what's bad about radio tracer imaging? The need for radioisotopes. You know, that's really a big barrier, and it's non-trivial because you've got to be thinking about, you know, having a cyclotron, having a radiochemistry lab, about all the containment measures that have to be taken to do both in vitro and in vivo studies. I mean, the animals pee after you've given them radioactive tracers, and you know, dealing with all of that can be logistically painful. And then the other aspect it's important to consider is that the NIS cDNA is 2 kilobases of extra sequence that you're going to have to add into your vector unless you use a separate vector to deliver the NIS transgene. And that, if you want to go clinical with that kind of product, then you have to ask yourself whether you're going to keep the NIS gene in the product all the way through FDA approval, or whether you're just going to use a product with a NIS gene in it for early stage development and then switch it out later. So these are some important considerations. So what is the NIS reporter? NIS is naturally expressed in the thyroid gland, where it concentrates iodine into the thyroid follicular cells required to make thyroxine. Been used forever for imaging and treatment of thyroid disorders, and is now used as a reporter gene, as you can see in that far right image, in which a mouse has been transplanted with NIS-positive bone marrow, and you can see the foci of NIS-positive bone marrow seeding into the bone marrow in the animal. We have multiple radio tracers available for NIS imaging. You know, it's a nice thing about the NIS Symporter that it was evolved to recognize and concentrate iodide, and we have multiple iodide isotopes that can be used. So I-125 has got a long half-life. It's got a gamma emission that's ideal for SPECT imaging in mice, so it's really convenient. For humans, that long half-life's a problem. So you got I-123 with a much shorter half-life and a good gamma emission. For human, I-124, the positron from that is a bit high energy, so you get blurry images, but you can use it for PET. And then I-131 is a beta and gamma emitter, so you can use that for destruction of tissue that concentrates iodide. But the NIS Symporter recognizes more broadly other monovalent anions. And so pertechnetate and tetrafluoroborate come into their own there because they both have a single negative charge and they're efficiently concentrated by NIS. And pertechnetate is made in many clinical imaging facilities. It's used routinely for imaging bone. And so it's convenient, readily accessible, short half-life, good for human and for mouse imaging. And then tetrafluoroborate uses F18, which is used for FDG glucose. So if you have the ability to make the F18, then with a radiochemistry lab, you should be able to make tetrafluoroborate. And that has a half-life of only 2 hours, so it needs to be used kind of on-site where it's made. Okay, now I'm going to walk through some stories, and I'll start with AAV, which is the current workhorse for in vivo gene delivery in the whole gene therapy field. And just, you know, this field of gene therapy, I should say, and I meant to say this at the beginning, it's transformative for the whole of medicine. I mean, you know, it's like the advent of surgery was 100 years ago. The advent of these genetic therapies is one-and-done treatments for diseases that otherwise needed kind of lifelong continuous therapy. So we've got CAR T cells, we've got corrective gene therapy for sickle cell anemia, thalassemia, for a whole lot of other bone marrow disorders. We've got gene therapy now for hemophilia. There was just an approval of a gene therapy for Duchenne muscular dystrophy, although that one's not working so well yet, but you know, it's the first step. So these are completely a new game in medicine. And there's a huge number of therapies coming through the pike. So I think this is important stuff. So we took an AAV9 vector encoding the NIS reporter under control of a CAG promoter, administered it intravenously to mice, and then on day 23 looked at the biodistribution of gene expression by imaging. Day 24, euthanized the animals, took tissues, and the idea here was to see whether the imaging correlates with the immunohistochemistry. So you can see from the images on the top here that there's really very nice transduction of the heart, the liver, the brown fat, and less so of skeletal muscle. Whereas in the bottom image, which is the control saline-treated mouse, what you see is just bladder, salivary glands, and you can't see the stomach in this animal because we've given it bismuth sulfate to mask the radioiodine in the stomach. If we look at the rotating images here, then you can see it really beautifully that you've got heart, liver, that brown fat pad on the back of the neck, and then skeletal muscle. And surprisingly, there was some degree of uptake in the gonads, which we hadn't expected, but there it is. And then when we did the tissue immunohistochemistry and compared, what you can see here is that it correlates really quite nicely with that in vivo imaging, and there's very strong uptake in the heart, in the liver, less so in the skeletal muscle. Interestingly, there's pretty strong uptake in the adrenal gland, very little in the spleen, and then there are indeed NIS-positive cells detectable in the gonad. So we then moved into the dog and worked with Maggie Redfield at Mayo Clinic, who was studying a model of restrictive cardiomyopathy due to cardiac fibrosis in a dog. And she wanted to be able to deliver genes into the dog heart. Thought that would work very well with AAV9 based on what we're seeing in mice and rats. But as you can see on the right hand here, the best imaging we could get in dogs following intracoronary infusion of vector was really poor. So she then started doing direct injections into the heart of the dogs. And the imaging was really very important here to help her to improve the injection technique using this spiral C catheter. And you can see that as she went through, additional dogs learning as she went from the imaging studies. And by the way, these images don't change over time in the dog. Once you've transduced heart muscle, it seems to be stably expressing the transgene in the same locations for months thereafter. So she was able to get to a much higher efficiency of transduction. Okay, what about oncolytic viruses? So this is how they work. The oncolytic virus infects the tumor, it spreads, it kills tumor cells, that recruits the immune system in, and the immune system damages the tumor. So you have two phases of tumor killing. You have the virus itself, the oncolytic phase, and then you have the immune phase. And the first oncolytic we started working with was measles, and we introduced the NIS gene into the measles virus, and then we found that that virus was effective in certain mouse models, and if we used the NIS reporter gene imaging to track it, we found that it was very selectively amplifying in the tumor. Actually, it's not amplifying a great deal here between days 2 and 5. But and you can see, if you look carefully, the distribution of the infected cells is more around the surface of the tumor than deep in the interior of the tumor. But nonetheless, we're getting good infection, and we're getting additional information on the distribution from those images. We took that virus into clinical studies, and we've done many clinical studies. The bottom line with those is that we've kind of stopped using measles because most of us have anti-measles antibodies, and they're really quite restrictive to the virus in human subjects. But here's a patient with breast cancer who happened to have no anti-measles antibodies, and when we injected the virus into a breast tumor in the axilla, shown here, that's the day 3 image. Sorry, the label seems to have gone, but the day 3 image of that tumor shows very strong uptake of radiotracer. And this was pertechnetate SPECT imaging. By day 8, that signal is fading, and by day 15, it's gone. But most importantly, if you look at the tumor itself, it hasn't really been hurt by the measles therapy. So measles got in there, infected cells, gave us a nice image, went away, and left the tumor unharmed. So there's this dissonance between tumor infection and the anti-tumor activity of the oncolytic virus. This is further illustrated here in a patient with multiple myeloma who got intravenous MV-NIS. And the SPECT-CT imaging shows that, well, first of all, the FDG PET image here shows where the tumors are. So the bladder is at the top, and these are massive subcutaneous plasmacytomas in her legs, big, bulky tumors. And if we look at the day 8 SPECT, the I-123 SPECT, you can see that there is indeed uptake by some of these tumors of the iodine-123, but it doesn't really correlate with the intensity of glucose uptake on the FDG PET. So there's clearly some heterogeneity in the uptake and infection of tumors by this virus, just shown in this one patient here. And everything fades by day 28 post-therapy. If we look at this is the same patient, and this is some pelvic cross-sections of the PET-CT and the SPECT-CT. And you can see on the PET that there are, you can see on the PET that there are 2 lesions. There's a subcutaneous lymph node in this section, inguinal lymph node, and then there's a deep pelvic lesion on the other side. And on SPECT, they're negative at baseline. They light up day 8, but the deep lesion is much brighter than the surface lesion. And then they're all faded by day 28. She had a partial response to therapy, but her disease relapsed fairly quickly thereafter. The other important insight from these studies with NIS imaging was that we could see a really powerful response of the virotherapy without necessarily seeing NIS imaging. And this was our star patient, Stacy Erholtz, who had complete remission of her myeloma after intravenous MV-NIS therapy. She had basically highly resistant multiple myeloma that was rapidly relapsing, was resistant to all available therapies. She'd had 2 stem cell transplants. And you can see at the time of relapse, she had this CT rendering. She had a big tumor on her forehead that was destroying the underlying bone and compressing the underlying brain. And she had the single infusion of virus and had complete remission at all sites of disease, but very weak NIS imaging signal, despite the fact that these tumors resolved. So, what this told us was that oncolytic virotherapy can actually be curative. And this woman's now 10 years out. She just celebrated her 10 years anniversary, still in complete remission, off all therapy. But the efficacy was likely due to immune control after infection of a small number of tumor cells, rather than massive infection of the tumor leading to its destruction. So we then move on to our next virus, VSV, which, importantly is low human seroprevalence. So we can use this to get around the problem of anti-measles antibodies. And we inserted a NIS transgene into this virus, as well as the interferon beta transgene, which is to try and enhance its ability to inflame tumors. So that NIS imaging has been helpful preclinically to help us understand the mechanism of spread of this virus. So if we look here at a study where we're taking mice with subcutaneous tumors, single intravenous dose of virus, and then daily NIS imaging thereafter to look for the uptake of the NIS tracer. You can see that if you compare day 1, day 2, day 3 in the same animal, you've got this advancing infection with increasing number of foci and increasing expansion of those foci, and that's borne out by the histological analyses that we've done. Very effective against these tumors, but not infecting every tumor cell. And here's a close-up of those day 2, day 3 images, just in the same animal. And you can see how the infection is actually spreading between days 2 and day 3 with enlargement of existing foci and the appearance of new foci, but there are still these massive voids of uninfected tumor, which somehow does disappear. And so the resolution, I think, does recruit the immune system, and that's a necessary part of the response. So we moved this virus into human clinical studies, and we did do imaging studies associated with the first few patients, but they were by and large negative. And it's pretty costly doing SPECT-CT imaging repeatedly on patients enrolled in clinical studies, and finding that kind of money was difficult. So we decided to leave it out for the study we did in hematologic malignancies. But what we found was that at the top dose level in our hematologic malignancy study, we had multiple patients with T-cell lymphoma responding. And interestingly, this inclusion of patients with T-cell lymphoma was based on a dog study that we'd previously done, where we had a couple of dogs with T-cell lymphoma that responded. And so with 50% response rate in patients with T-cell lymphoma, we've now moved into an expansion cohort. But these are not NIS images. These are FDG PET images of before and after therapy in patients with T-cell lymphoma getting a single intravenous infusion of the virus. And you can see there are really some quite dramatic multifocal responders here to the systemic virotherapy. So we're excited about it. But the question of the NIS imaging is kind of interesting, because we've still got the NIS gene in this product that we're developing and that we would hope someday to get approved. But we're not actually using the NIS imaging now as part of the product development. If it did get approved, it would open up a lot of possibilities for further exploratory NIS imaging studies in human subjects. So we'll see. All right, so let's move to the rhesus iPS cells. So here, the big question was a kind of emperor's new clothes question. So lots of people are saying, okay, we can repair the heart with, lab-cultured iPS-derived stem cells, and there was little evidence that those cells actually were integrating in, persisting in the hearts that were supposed to be repaired. So Cindy Dunbar worked with us and decided to use NIS imaging to try and address this. So she made rhesus iPS cells, edited them to express NIS, not just in the iPS cells, but in their differentiated progeny. The NIS-positive stem cells did concentrate radioiodine. It was inhibited by perchlorate, as you'd expect. And then she used this rhesus monkey cardiac injury model in which myocardial infarction was induced and then repaired with the rhesus NIS cardiomyocytes. And the imaging studies you can see here from sham versus the stem cell treated animals show this focus of activity in the muscle where they injected the NIS-positive cardiomyocytes. And then at the time of animal sac, you can see that the sections of the heart from this area show very convincingly that there are a population of NIS-positive cells there, fully integrated into this functioning cardiac tissue. So what about lentiviral vectors? These are the workhorses of ex vivo gene therapy. They're used to generate cells that are used for CAR T therapy, for corrected bone marrow stem cell therapy, and preclinically, at least up to this point, quite a lot for the regeneration of organs such as the liver. So we worked with Ray Hickey and Scott Nyberg to look at whether we could track liver regeneration in mice and pigs. And this was a model. It's hereditary tyrosinemia, which is due to a knockout of the FAH gene. And interestingly, these animals become dependent on a drug. They need nitisinone drug therapy in order to stay alive. And if you withdraw that drug, their liver just degenerates. So what you can do is you can infuse into these animals or inject into these animals normal or corrected hepatocytes, withdraw the drug, and the good hepatocytes will repopulate the liver. So we decided to check out whether we could non-invasively track the liver regeneration using two vectors, one encoding the FAH, another encoding the NIS cDNA. And mouse hepatocytes obtained from FAH-negative mice were transduced with the pair of lentivectors, then transplanted into FAH-negative recipient mice, drug withdrawn, and serial imaging done. And you can see from these liver histology slides that eventually, from this protocol, you get really good repopulation of the liver with the FAH-positive cells. So these are images from an animal taken day 41 and day 85 after initiation of therapy, and you can see there are multiple foci appearing in the liver that are expanding and apparently increasing in number as well over time between day 41 and day 85. And if we, sorry, this is my Noah's Ark slide. It's animals coming in 2 by 2. So these are the control animals with FAH lentivirus only, no NIS, and these are the animals that got the NIS, and you can really beautifully see these nodules repopulating the liver. And you can see that it's spread throughout the liver and that it's not totally uniform throughout the liver, but nevertheless, it's nodular. And so you get a good feel for how this is actually happening. So we then moved it into the pig model because we had pigs that were FAH knockout. And the logistics of this is really quite painful. You know, Scott Nyberg, who's a surgeon, decided to create this pig model. The way you make the homozygous pigs is you breed heterozygous parents, and the mother would have like a litter of, say, 17 babies. And the danger with mother pigs is that they lie on top of their babies and they kill them, and then they eat them. And so the surgical fellows had to sit overnight watching mother pig to make sure that she didn't suffocate any of her babies and push her off if she did, because they knew that only 1 in 4 of those baby pigs was actually homozygous FAH knockout. And then having got such a pig, a liver biopsy sample is obtained, transduced with FAH and with NIS, and then reimplanted. And these are the pig images that we were able to get from that study. And again, the logistics of this was something else because pigs grow super fast. And note, this is 8 months, this lower set of images. And, waiting 8 months means the pig gets very big. So this was done at night on clinical imaging instruments, and it's a big logistical exercise to kind of get the pig. But the reason we didn't do any imaging beyond that date is because the pig's too heavy and big to actually allow the imaging to occur. But what you're looking at is not the bright image here, which is the stomach with some bismuth sulfate in it, which was supposed to hide the image. But you're looking at this, the liver, the paler blue signal on the lower set of images here. And what you can see from this is that there is nodular regeneration of the pig liver, and it's pretty uniform throughout the entire pig liver. And you can get at this by doing biopsies, and the liver biopsy here shows great repopulation of the liver with FAH-positive nodules, but really the image shows you the entire organ. Okay, so now I want to get on to the last thing, which is the use of NIS imaging to guide the development of a novel approach to CAR T-cell therapy. And this is something that's now very close to my heart because I'm very interested, and I'll explain why, in trying to deliver CAR genes direct to T-cells in vivo. So the process of CAR T-cell therapy is kind of painful. The patient has to get apheresis. The T-cells are sent off to a center where they're transduced, qualified, sent back, and then infused into the patient. In the meantime, the patient gets immunosuppressive chemotherapy to make room for those T-cells, and the whole process takes a few weeks. Then post-therapy, there's this high probability of toxicity, cytokine release syndrome, neurotoxicity, et cetera. And so patients have to get their treatment in certified medical centers, and they have to stay close to the medical center for 4 weeks. So logistically, it's a real pain. Now, CAR T therapies work really well. This is a patient with, late-stage treatment refractory multiple myeloma, single infusion of CAR T cells against BCMA, and 2 weeks post-treatment, the disease is gone. And these responses are durable. But only a small fraction of potentially eligible patients are actually getting their CAR T therapy in the US at the moment, because of high cost. And it ends up being about $1 million per patient because not only is the treatment expensive, but then you've got all these logistics surrounding it and the hospital costs and the toxicity management. The toxicities are frequent, so that's something that puts people off. And the logistics are challenging. The travel and lodging at the treatment center, finding a caregiver to accompany you, and the long wait times until the CAR T cells are ready to use. So if your disease is progressing fast, you're not really a candidate. So the vision we have for the company is to generate lentiviral vectors that can deliver the CAR direct to the T cells in the patient, in the body. And the idea of that technology is shown here. We have the lentiviral vector with the VSV G protein on it. We've been working for a long time on G protein engineering to display single-chain antibody on the G protein to retarget, and in this case it would be anti-CD3, to engineer the G protein to ablate its natural tropism, and to introduce resistance to serum neutralization. So that technology is working pretty well now. And just to show you some targeting, this is a display of an N-terminal CD3 antibody on a blinded protein. We've got Jurkat cells here that are CD3 positive, Raji cells CD3 negative. You can see the wild-type G protein gets you into both cell types, the blinded vector into neither. And if you target CD3, you get very efficient and selective transduction of the Jurkat cells. So we then, we've done a lot of studies with this, but I just want to show you whole blood, because I think whole blood's a really, really kind of critical system to be able to work with. Because in blood, 1 ml of blood contains a huge number of red blood cells. It contains a large number of platelets, leukocytes, and only 1e6 T cells. So there are very few T cells, and what the virus has to do is to be able to pick out those T cells specifically. And also there's a lot of antibodies and complement in blood that can destroy the virus before it gets access to the T cells. So what we did was take blood from volunteers at the lab. We took fresh blood, immediately plated it, added the virus to the blood, left it for 6 hours, and then separated out the leukocytes, the PBMCs, and grew them without the addition of growth factors that are known to stimulate the growth of T cells. So we just let them go. And what we were looking for was to achieve greater than 10% T cell transduction with a high level of specificity. And these data, I'm telling you, for those of you who haven't looked at this kind of thing, you probably don't realize, but this is, to me, staggering. I mean, this efficiency of targeted transduction in whole blood. So with no lentivirus, we're seeing no GFP-positive cells. With a wild-type G protein, we're seeing no GFP-positive cells. And with the CD3-targeted virus, we're getting this big population of GFP-positive cells, and they are CD3-positive, they're T cells. And they increase in number between day 4 and day 6. And by day 6, in this first patient, we've got about 6% of the T cells transduced. In the second patient, we've got 13%. So we've exceeded our 10% goal. So then we did some studies in mice that were reconstituted with the human immune system. And you can buy these mice, they're expensive. You can buy them when 40% of their blood cells are actually human. And then you can give them intravenous targeted lentiviral vector, and you can look at them by imaging, checking their blood periodically. And we sacked these animals day 39. So I'm just going to show you day 39 blood data. And when we use the CD3 targeted virus, we see this population of CD3 positive, GFP positive cells. They're transduced T cells expressing a CAR. And we don't see them with wild type, and we don't see them with saline. But then if we go down and we look at the B cells, so here we're staining up the y-axis with CD19 to look at B cells, there's a big B cell population that's looking quite healthy in the saline and wild type groups, but it's gone in the CD3 targeted vector. So these T cells are wiping out the B cells because they're targeting CD19. And if you look at the tissues in these animals, the bone marrow, the spleen, and the liver, you again see this population of transduced T cells in every case. And in every case, you see that the B cells are very efficiently depleted. So this really is in vivo generation of CAR T cells that wipe out the B cells. So it's an exciting development. Now, what about imaging? So we've worked extensively with Saad Kenderian and Leo Sakamura at Mayo Clinic, looking at introduction of the NIS gene into CAR T cells. And so here we have a CD19 and a BCMA CAR, and we co-introduce the NIS using a different lentiviral vector. So you can get CAR-positive, NIS-positive cells. And if you implant those cells subcutaneously into mice, this is a 4-quadrant experiment where we just put a different dose into each quadrant. And what you see is that with the top dose level, you get a stronger signal than you get with the lower dose levels, and eventually you get below threshold. So we know something about that. The CAR T cells show potent efficacy even when you've introduced the NIS gene. So here's just treating the OPM2 myeloma model with BCMA CAR cells that have the NIS transgene in them, and compared to the wild type cells, you get good tumor response and prolonged animal survival. So these CAR T cells work. And if we look at the images on day 8 post-therapy with the CAR T cells, in the control mice, you don't really see anything other than the thyroid, stomach, bladder. But in the BCMA CAR HNIS T cell treated mice, we see this very nice signal appearing in the bone marrow, which is where the tumor cells reside in this myeloma model. And if we clean up those images, you can see very nice bone marrow transduction here, and in the other images in this picture. And if you look at what happens between day 8 and day 15, what you see is that this strong signal, which comes from the T cells attacking the tumor in the bone marrow, actually diminishes greatly by day 15. And by day 15, there's very little tumor left in these animals. So it's a dynamic process where the T cells are going to be at their zenith, at the time that they're attacking the tumor, but the tumor is going to disappear and the T cells are going to fade thereafter. So this is a situation where you really need good dynamic analysis of what the cells are doing. Using NIS imaging, Saad also did a study in order to see if there was a difference in what he could see in animals that had cytokine release syndrome versus those that didn't. So here he's implanted patient B acute lymphoblastic leukemia blasts into animals and then given them CD19 CAR T cells with the NIS transgene, monitor the animals for symptoms of cytokine release, and do the TFB PET CT imaging. And the results are really quite striking. When there's no CRS, what you see is just a spleen signal, because the T cells are attacking the tumor in the spleen. That's the main site of tumor growth, whereas in this particular case. Whereas in the case of animals with CRS, you get the T cells all over the place. They're in the spleen, they're in the lungs, they're in the liver, and doing a lot of damage to the animal. What about this in vivo obsession that I have? You know, this is very hot off the press, so all I've got is something really preliminary here, and I'm kind of going to finish up on something that's less compelling than what I've shown you up till now, but I think nevertheless it's pretty good. So, you know, here we're using the fully retargeted lentiviral vector encoding a CD19 CAR. We're giving mice B-cell lymphoma using the NALM6 luciferase cell line. And then just one day after we've given the tumor, we're giving human peripheral blood mononuclear cells, and then we're chasing those with lentiviral vector to transduce the cells in vivo. And then we're doing imaging a week later on day 8 for luciferase, day 9 for TFB, and then again on day 14 and day 15. And so what you can see here is that in this animal, and this was a very small pilot experiment. So in this second animal shown here, on day 8, we've got a very strong luciferase signal indicating there's tumor all over the place. And then by day 14, that signal is gone. So the in vivo generated CAR T cells are attacking the tumor. But can we see anything corresponding to that when we try and image the T cells? So this is a control animal where, you know, we just see what we'd expect to see: bladder, stomach, thyroid, and you can see the salivary glands in this case. And when we look at that mouse that I was showing you that had a lot of tumor on day 8 and very little tumor day 14, this is the day 9 TFB PET. And what we see is a signal here: focal uptake in an axillary lymph node, focal uptake in a thoracic nodule, diffuse uptake in the spleen, and diffuse uptake in the liver. And we see, although it's a weak signal, diffuse uptake in the spine here. So I'm pretty certain that we are seeing these CAR T cells acting against the tumor in vivo. I feel we've got our foot in the door and that we can really use this technology as we go forward. So to sum up and conclude, reporter gene imaging is key to pharmacologic characterization of genetic and regenerative medicines. Main barrier to reporter gene imaging using NIS is the radioactivity barrier, which is not trivial. Many insights have been gained from preclinical clinical studies. I've told you about AAV vectors to study biodistribution and durability of expression. Oncolytic viruses, how we've got a handle on the relative contributions of the oncolytic and the immune effects. iPS-derived stem cells, that they really do persist at sites of implantation and participate in tissue repair. The uniformity and nodularity and kinetics of liver regeneration with NIS-positive hepatocytes, and the complex dynamics of fluxing CAR T cell interactions with fluxing tumor cell burden. And I'm really keen on working with the NIS system to further understand what's going on dynamically in this direct in vivo gene delivery system. So the best imaging agent is TFB PET. It gives us the highest resolution imaging, NIS imaging that we can get, but it's not yet clinically approved for routine investigation of human thyroid disorders. If it were, it would be much more readily available in those centers that do have a cyclotron and in those centers that actually make fluorodeoxyglucose for PET imaging. So, you know, if that comes to fruition, then I think it could be quite a game changer for us, because the TFB images are superb. And just to, you know, just to make the point that human studies with TFB are happening. These are TFB images generated at Mayo Clinic by Tim DeGrado's group. These were published a few years back, actually, but you can see they're absolutely beautiful with the salivary thyroid, stomach, bladder, very nicely lighting up with very acceptable background. And you know, if this does get approved for clinical use, then I think it's going to be much more readily available to a lot of investigators in centers that have clinical radioisotopic imaging going on. So I'd like to finish with acknowledgements. There's a lot of imaging people at Mayo Clinic who work closely with us, especially Tim DeGrado and Val Lowe. A lot of clinicians have worked with us on the different approaches that we've discussed here. I need to call out Ka-Wai Peng, who's here in the audience, because every single one of these studies that I've showed you, she's been at the heart of it. She's been the kind of enthusiastic driver, jumping up and down, making sure the logistics get dealt with appropriately and that people stay on target, that images do get processed and analyzed at the end of the study. And of course, the patients and their families who participate in the clinical studies. So I'll end there, and thanks very much for your attention.
Steve, I would like to thank you very much for this outstanding lecture, outstanding results, and great presentation. Thank you. So what an example of a off-the-shelf, genetically encoded biological, ready to go. Thanks. Imageable biological, ready to go. Okay, so we have maybe time for one or two questions. If anybody would like to approach the microphone, please.
Great talk. Just from imaging point of view, do you compare the NIS imaging compared to early use luciferase or GFP, especially for AAV9? You know, I know the NIS has the advantage of less immunogenicity, you know, more 3D imaging, but in terms of the ratio of broad biodistribution, I think it should be consistent. Second question, can you comment on this NIS imaging for no viral lipid nanoparticles tracking is for early discovery project?
Yeah, so yes, from what I can gather from the literature, the biodistribution of gene expression with luciferase and GFP using AAV vectors is the same, as you would expect. I think the durability of gene expression from the luciferase and GFP vectors is significantly lower because they are immunogenic, you know, and if you're doing a study in an immune-competent animal, you start losing those transduced cells over time. As far as the nanoparticle approach is concerned, I really have no experience of that. I've been focused pretty much entirely on the viral gene transfer systems. I know that Christine Spitzweg, who used to be at Mayo and worked closely with us, is now kind of really pushing nanoparticle-based NIS imaging, and I think is making good headway with it. So it has potential there for sure. It's all down to what's the efficiency of gene delivery that you can get in order to get, and what's the sensitivity of the imaging equipment that you've got in order to detect the signal.
Okay, thank you. Please, the Tsien first.
Do you have a hypothesis for why you observed the unexpected uptake in the gonads when using your NIS AAVs?
We have not dived deep into that. I mean, you saw from the immunohistology that I showed that we should be able to identify the cell type that is transduced in the gonad, and it does not appear to be the germ cells or the germ cell lineage. So, that was a kind of reassuring point about it. But we were really doing the study just to kind of do the comparison between the technologies rather than we weren't actually ourselves developing an AAV therapy at that time.
Thank you. I'm sorry for the, in order to conserve some time, one last question, Tim, if you don't mind.
No.
Okay, on this side, please.
All right, thank you. One quick question. I actually have a few, but I will just focus on that one then. You mentioned in the beginning of your talk the immunogenicity of NIS proteins from different species, and you also mentioned the minke whale, which you kind of are interested in. Did you ever test for immunogenicity in different organisms of the minke whale NIS? And if so, and you do not see immunogenicity, do you see any potential of using that as a reporter gene in human trials?
We have not formally tested. What we've done is to look for long-term gene expression in immune-competent animals without attenuation of signal. And we've seen that with the minke whale NIS protein. So that leads us to believe that it's not immunogenic. And so that's kind of the basis of my assertion. But we haven't developed multiple analytic methods to be able to identify hidden, so we're not detecting T cells against NIS or antibodies against NIS. We're just seeing the durability of expression independent of the origin of the NIS protein. And if you have other questions, I'm going to be around after this. Very happy to.
Yeah, I'm trying to find you. Thank you very much.
Yeah.
Okay, let's thank again Professor Russell, and that was great. Thank you. Okay, so we conclude our last plenary for the meeting. We have a break, and then the program continues. Thanks.